So I'll let you know when you start your slides.
Yeah.
So if Alan starts a live stream, oh, excellent. Good morning, everyone. Welcome to session 6 on exceptional preservation. So we've got 6 talks, and we're going to start off with Morton in the Cambrian. Morton, if you could share your screen.
Yeah.
Excellent. Great. And you, yes, well, let's see.
All right, good morning. Today I'm going to talk about how the series placid Lagerstätte can help us better understand the phosphatization of soft tissues. So phosphatization is the secondary replacement of tissues by apatite, which is a calcium phosphate mineral, and it's able to preserve fossils in the most amazing details down at the subcellular levels. So here we're going to focus on the internal phosphatization of labile soft tissues such as muscles and digestive systems. And this type of phosphatization is relatively common in the fossil record and can even be created in laboratory experiments within a matter of a few weeks or to months. And so far, available data has shown that it's very biased in what it preserves where. And part of this bias is because that it is very dependent on a decay-induced chemical microenvironment and the availability of phosphorus. So there's things that we still don't know. And that is why it appears that different tissues or different taxa preserve at different localities. So one of the issues is that often the amount of phosphatizations is very low at these deposits, despite them being relatively common in the Phanerozoic. But all the phosphatization is restricted to only a very few species, and that makes it very difficult to quantify the community scale preservational patterns, and therefore to find commonalities in phosphatization. And without these commonalities, it's very difficult to understand the mechanisms for phosphatization. But fortunately, we have the series placid Lagerstätte from the early Cambrian of North Greenland, which is a natural laboratory to study phosphatization. And that's because here we have very high abundances of fossils, and they show a high frequency of phosphatization in a range of different species. So this plot here illustrates just how dense the fossil abundance are in series placid compared to Burgess Shale and Shenyang. And not only do we have a lot of these fossils or high density of fossils, we also have a lot of fragmented organic material seen on this slab here. It's all these gray patches. But we also have the occasional microbial mats, as this smooth surface in the upper left corner here shows. So all these phosphatized tissues can be used to reconstruct the internal anatomies of early animals, as for example, the digestive structures seen here in some panarthropods. And this holds both ecological as well as phylogenetic implications. And we can do the same for the muscle anatomy in a range of different taxa as well. And this has already been done for the gilt lobopod Pampdelurian by Young and Wind in 2016. But here we're going to focus on how we can also better understand the mechanisms for phosphatization. And this is part of my PhD. And for these preliminary results I'm going to show here, we've used the published records of phosphatized tissues and then also a quantitative database of which tissues preserve in which species based on the 2016 collection. And here we have over 800 phosphatized specimens. And so the ultimate goal is to understand, based on the series placid material, why and when certain tissues phosphatize in certain species. And then hopefully this can also help us understand the biases that operates in other Lagerstätte as well. So now I'm going to show you the distribution of the different tissues in series placid. So this is an overview of the diversity in series placid. And then I can plot which tissues preserve the digestive systems and to what extent. And then I can in turn plot the muscle tissues that are preserved in the different species. And then lastly, we have some other tissues, such as the ventral ganglia and stem ketogates that Jakob talked about yesterday, which is on the far right. So as you can see here, there's a high variability in which species preserve what. There are some species that just never preserve anything or only preserve some of the gut structures. So what controls this preservational variability? Well, first of all, it's very complex and there are a lot of different variables. But the preliminary results that I'm going to show here, they can reduce some of this noise, and then we can get closer to what the important mechanisms are. And here we'll focus on three particular factors. It's the phosphorus content, the size, and the microenvironments. So all the tissues, or at least the most common tissues to phosphatize in serious pests, they're all pretty rich in phosphorus. It's digestive systems, muscles, and these ventral ganglia. But sometimes the muscles are restricted to the immediate vicinity of the gut diverticulae, as seen on this illustration here, where muscle is the red and occurs as patches around the diverticulae. And diverticulae being these pouches that sits on the digestive tract of the gut. Sometimes the patches are a bit larger and it becomes this confused mass around the diverticulae in the axial region. And diverticulae has been proposed to be especially rich in available phosphorus because they might have function as storage facilities for phosphorus and calcium ions. And we see the same pattern with these ventral ganglia and stem ketogates that which are also rich in phosphorus because it's where the nerves concentrate the cell nuclei, which in turn are rich in phosphorus. I have phosphorus-rich DNA. And here on the far right, we see that the muscles also occur where these ganglia are expected to be. So this means that not only do phosphorus-rich tissues have a higher potential to phosphatize themselves, but they can also augment the phosphatization of the adjacent tissues. But we still see that not all phosphorus-rich tissues phosphatize all the time, which is shown here by the arthropod Campana. So what explains this variability? Well, one factor that can do that is size, because we noticed that small specimens or species appeared not to phosphatize as much as the large ones. So if we use Campana and plot the type of phosphatized tissues against the width of the specimens, we see that digestive structures are phosphatized in even the smallest specimens we have. But on the other hand, muscle preservation does not kick in before they reach a certain size. In this case, about a width of 20 millimeters. So that means that size introduces a taxonomic as well as an ontogenetic bias to phosphatization. And another thing that can impact this variability is microenvironments. And we can test that in serious pests because we find that Campana, our charismatic model organism for phosphatization here, that species is sometimes found in an enrolled state. And when you're an arthropod and you're enrolled, you create a very effective barrier to the external environment. And this means that you're able to sustain a closed environment inside yourself. And when you're decaying, it becomes this microenvironment, this decay-induced microenvironment, which favors phosphatization. And what we see in the collection is that all the specimens that are there, they actually show some degree of phosphatization. But not only do they show some degree of phosphatization, they actually generally show a much higher frequency of the most phosphatization that we have. In this case, represented by this lateral tissue here that we only see when we have the most extensive type of phosphatization. So From serious passage, we have identified these three factors that the data suggest are important. It's phosphorus content, the size, or perhaps anatomy, and the microenvironment. However, it's important to stress that these factors do not favor phosphatization, or they do not initiate phosphatization on their own. Instead, they need to work in concert. And just to illustrate this point, I've here made a conceptual diagram where this circle represents all the different taxa and the tissues of serious passage. And within this circle here, all the taxa that occurs within this blue circle, those are the taxa or tissues that have enough phosphorus content to phosphatize. Then the red circle here are all those taxa and specimens that have the right sizes to phosphatize. And these are those that have the right microenvironment to phosphatize. And what we suggest is that these three factors need to overlap to initiate phosphatization. So it's only all the taxa that occurs or tissues that occurs within this green field here that actually phosphatize. And this diagram here is for a depositional environment which favors extensive phosphatization, where there is a lot of decay in organic matter in general, as in serious passage. In other environments, it might be that there's a much smaller overlap between these three factors, which means that there are less species within that overlap that can actually phosphatize. For example, the Burgess shale, where it's only mainly the gut diverticulae which phosphatizes.
That's 1 minute left. Oh, perfect.
Perfect. So to conclude, phosphatization is a very complex and biased process, and we still don't know all the factors that can influence it. However, the preliminary data that I've shown here allowed us to get a little closer to the important factors for phosphatization. In this case, phosphorus content, size, and microenvironments. And there's a lot of work to be done still, and that's some of the things I'm going to work on as part of my PhD here. Thank you very much.
Excellent, thank you, Morten. So chats to Morten in the Slack, and we can talk about them at the end. In the meantime, Alison, if you could unmute yourself and share your slides. Thank you, Morten. Excellent, Alison. So we can see your slides. We can't see.
Sorry, that's okay. Now you can hear me.
Excellent. So Alison's going to be speaking to us about soft tissues in vampire squid. I believe, take it away. Thank you.
Awesome. Yes, good morning, everyone. I'm Alison. It's lovely to be here at PalAS, albeit in a slightly virtual setting, virtual sense. Oh gosh. Okay, hang on. Bear with me. Okay. So the overarching questions in my PhD all deal with understanding or better understanding the relationships between fossil and extant coleoids. So as you can see, coleoids originate in the late Paleozoic. They diversified in the Mesozoic, and today their modern forms, squid, cuttlefish, octopus, occupy nearly all marine habitats. But their evolutionary history is not well known. And very simply put, this is due to the lack of comparative material. Basically, soft tissues rarely preserve in the fossil record. So my current research is focused on Vampyromorpha. This is a clade that includes Vampyrotoothless infinalis, this rather mesmerizing creature here, and their fossil relatives. So Vampyrotoothless is the only extant vampyromorph, and their phylogenetic position has been heavily scrutinized as they exhibit several autapomorphic characters. They also have a mode of life that is unique among modern coleoids. They occupy deep sea environments, often found in and around oxygen minimum zones, and they're detritivores. But today, though, they are accepted to be positioned within the 8-armed Octobrachia up here. So for my study, I'm using vampyromorph coleoids from La Vaute-Saronne. This is a Lagerstadt in the Ardèche in France. It's mid-Jurassic in age. And this site is unique as it preserves specimens in 3D. And you can see some of the samples from some of the Jurassic coleoids from the locality. Most specimens recovered from this site were described in the 80s by Fisher and Ryu, although in recent years some have undergone reappraisal. In 2002, Fisher and Ryu described a series of 8 specimens from this locality and assigned them as a new species, Vampyronassa rhodanica. And based on external observations, they positioned them as the oldest likely relative of Vampyrotoothless infinalis, our friend over here. And again, this is the only extant vampyromorph. The only other species that has been designated a fossil relative to Vampyrotoothless is Necrotoothis, and this is from the Oligocene. However, it's only known from its gladius, the internal shell, but it has been described recently by Kostak et al. 2021 as having the same deep sea mode of life as Vampyrotoothless. So the gladius is the main feature in fossil coleoid systematics. As I mentioned, soft tissue typically doesn't preserve. So the gladius provides the character state data. And as Fisher and Ryu weren't able to comment directly on the morphology of the gladius in Vampyronassa, the relationship is not yet widely accepted by the fossil cephalopod community. And in a recent synthesis of Octobrachia systematics by Fuchs 2020, Vampyronassa were determined to be Vampyromorpha incertisedis. So this brings me to my current research. By reappraising Vampyronassa, am I able to confirm the position of Vampyronassa as a Jurassic Vampyrotoothless relative? And what can I learn about the ecology of this Jurassic form? I initially began working on these 4 specimens. All were ascribed by Fisher and Ryu 2002 as Vampyronassa rhodanica. I wanted to have a larger data set for morphological comparison. So from the 3 fossil suborders, I identified 13 potential contemporaneous specimens from either the same time bin or locality. However, many lacked information on directly comparable characters. So my resulting sample is a bit smaller than I'd hoped, but it includes 4 fossil genera, all from the Levault locality, and 4 extant genera, 2 Vampyrotoothless, an Octopoda, and 2 Decabrachia. So to gather new information on the Vampyronassa specimens, I used X-ray-based imaging techniques. And this allowed me to observe previously unseen internal organs, as well as get better resolution on external soft tissue. And in the fossil specimen that you can see on the top, the grayscale contrast reflects the density of the mineralized soft tissue. The extant specimen on the bottom, Vampyrotoothless, was stained prior to scanning so that the contrast of the soft tissues would be amplified. And then the contrasting boundaries were used to demark the outlines of the various anatomical features for segmentation. Excuse me. So here are 2 of the reconstructed specimens, Vampyronassa on top and Vampyrotoothless on the bottom. Unfortunately, this technique did not allow me to identify or reconstruct the gladius of Vampyronassa. And without this, I wasn't able to confirm or deny Fisher and Ryu's assertion that they were relatives, although I was able to compare the soft tissues between the 2. Many of the internal organs were challenged to identify with any certainty. Obviously, there was some post-depositional squishing happening. So I focused mainly on the arm crown, as this was the best preserved element across the fossil specimens. So question 1. There are currently 2 published studies that combine morphological characters of both fossil and extant coleoid taxa. And Vampyronassa has been included in both of these studies. This is one from Kruta et al. 2016. And you can see it returns a topology where the 2 species are sister taxa. Using the soft tissue data that we gathered, we were able to resolve some characters within this existing phylogeny that had been previously been question marks. One key finding was that Vampyronassa rhodanica had the type of sucker attachment that is unique to modern day Vampyroteuthis. And the presence of this Vampyroteuthis-like attachment in the Jurassic was previously unknown, and so we were able to add it as a character state for the first time. Additionally, we were able to determine that Vampyronassa suckers had radial symmetry, and this is known commonly in octobrachia. We can see that the sucker stalks are present, but not clearly attached to the arm muscles, and you can see the sort of like ghost-like shading almost. And there's no evidence of a sucker lining, which is a feature seen in some decabrachians, and you can see it here in an extant loligo. So we incorporated the updated character states in the matrix of Kruta et al. 2016 and reran the tree, and there were no changes. So our data supported the position of Vampyronassa and Vampyroteuthis as sister taxa. Although obviously we were only able to resolve 4 character states out of the 132, and more than 50% of the matrix is built on gladius morphology. So as we don't have this element, we're sort of taking our results with a grain of salt. And on to question 2, we can see that the dorsal arm pair in Vampyronassa is more specialized. They're longer than the other arms, and the suckers and ciria are located only on the distal portion. Excuse me, the Vampyroteuthis-like suckers are, sucker stalks are slightly more elongated compared with the other arms. However, they're not elongate enough to be comparable with decabrachia. And additionally, they protrude into the muscles at the base of the sucker, so the acetabulum. And in modern forms, it has been shown that this kind of insertion functionally is more resistant to pull force when the sucker is active. In the other arms, the suckers are present all the way along, and they're proportionately more robust than those of Vampyroteuthis. The infundibulum, this area here that adheres to the surface is larger, and the stalk also protrudes into the muscles at the base of the sucker. This is the same in the sessile and the dorsal arms. In modern forms, the cirri, which are these fleshy protrusions here that flank the suckers, they're described as having a sensory function, and you can see that they're much more robust in Vampyronassa. So while Vampyronassa does have some characters that reflect those in Vampyroteuthis, they also have a mosaic of characters that suggest potential for increased predatory function. And in addition to the specialized arm crown, they also have a more muscular and streamlined mantle. So our interpretations. Today, the mode of life that is employed by Vampyroteuthis is unique amongst colioids, but how and when this ecology and lifestyle developed isn't clear. It is hypothesized that the ancestral form, Necroteuthis, was already exploiting this niche by the Oligocene, as discussed by Kostak et al. 2021. But our new soft tissue data does allow us to add more resolution to the vampyromorph timeline. We know, for example, that the Jurassic form had the Vampyroteuthis-like sucker, which indicates a potential link between the two species. Relationship-wise, the characters that we were able to add to the matrix of Kruta et al. 2016 supported the relationship between Vampyroteuthis and Vampyronassa. And from the soft tissue morphology, we can infer that in contrast to the lifestyle of modern forms, the deep sea dwellers, the adaptations seen in the Jurassic taxa are indicative of a more pelagic and predatory mode of life. Thank you very much.
Thank you, Alison. That was a really interesting talk with absolutely fantastic specimens.
I'm so lucky, they're beautiful. Yeah.
Yes, really, really fantastic. Excellent. Well, thank you. So next up we have Joe. So if you could share your slides. Morning, Joe. It's very early for you. Very much appreciate you being here and giving us a talk on marine arthropods from the Ordovician. If you could share your slides and take it away.
Thanks. It's nice to be here as early as it is. My name is Joe Moisiak, and I'm a PhD candidate at the University of Toronto and the Royal Ontario Museum. And for those of you who know me already, you may be surprised to see that today I am not presenting on the Burgess Shale, but instead on some interesting new instances of soft tissue preservation that we found in the Ordovician of southern Ontario. So we're all familiar with the famous Burgess Shale type deposits of the Cambrian period, but some of these deposits actually extend also into the subsequent Ordovician period. And these are really critical for telling us what's going on about this transition period between the Cambrian biotas and these unique soft-bodied organisms that occur there, and then the subsequent Ordovician and later Paleozoic times. And so one of the really critical sites from the Ordovician is the Feijoada of Morocco. And at the Feijoada, we see this really interesting mixture of Cambrian typical taxa like morellomorphs and radiodonts, and then mingling in with taxa that we associate generally more with the Ordovician. So things like horseshoe crabs and various diverse echinoderm groups. Now, we could question whether the Feijoada is sort of generally representative of this Cambrian Ordovician transition in terms of its soft-bodied faunas. And of course, it's only one site, but we do have some other sites from around the world that give us some similar glimpses into similar sort of open shelf environments during the Ordovician. But they are relatively sparse around the world. And so today I'm going to be focusing on some new findings from North America, from Laurentia. So we actually already know of some Lagerstätten from Laurentia that give us some hints about the soft-bodied biotas that were living here. But one thing to note about a lot of these sites is that they represent sort of weird environments. And so we have these relatively shallow, restricted environments, typically low in oxygen, high in salinity, and having a low diversity of taxa, so dominated by things like eurypterids, horseshoe crabs, and macroalgae. There are a few exceptions to these rules, and so the Beecher's trilobite beds is a very notable one. But even this is a little bit of an odd environment. So it's a relatively, or a rather deep water environment, low in oxygen, and representing this sort of distal turbidite facies. And so we could really question how much any of these sites are representative of what was generally going on in terms of soft-bodied biotas in open marine shelf environments that characterize large parts of what is now North America. And so in this talk, I'm going to be showing you some new material that we've found in southern Ontario, and specifically around Brecon, Ontario, which is a little bit east of Lake Simcoe in the south central region. And so we've been working with an amateur collector named George Kampouris, who's secured permission to work in some active quarries in this part of Ontario. And George has been focusing his work on some really remarkable assemblages that occur in the upper part of the Kirkfield Formation, which is earliest Cattian in age. And so this part of the formation is a carbonate dominated. So this is representing this wide open carbonate shelf environment, which has an incredibly diverse fauna. And so Timothy Patton and colleagues have already done some really nice work on the sedimentology of the upper part of the Kirkfield Formation. And so we're focusing on this little zoom in section on the left here. And generally what seems to have happened here is that we have this lower basal hard ground unit, which formed, it's very highly tiered. And so it's a very complex surface, and all sorts of encrusting organisms made their homes on this hard ground surface. And then repeated storm events buried that hard ground, and they buried it again. And each time entombing the communities that were living on this hard ground. And I want to draw your attention in particular to a bed that we call bed 3, which is a shale bed, where we found, in addition to exceptionally preserved shelly organisms, some soft-bodied macrofossils for the first time. And so here's just a photo of what the site actually looks like on the right here. And you can see one of these hard ground mounds, this M41 in the foreground, which is protruding up through some of the overlying beds. And so again, bed 3, which would have been blanketing the top of this large surface here, is the one that we really want to focus on. And so, like I said, we find this really exceptionally diverse shelly biota in the Brecon area. And so on the right, you can see one of these crazy mass assemblages of echinoderms, which incur this incredible density, especially around the hard ground mounds. But we have also found some soft-bodied fossils. And so we have things like macroalgae on the far left here, carapace fragments, and a new species of morellomorph, which I'm going to show in a minute. I should note that the soft-bodied fossils at this site are extremely rare so far, representing a very small fraction of the total number of specimens collected. But we think that there's probably some sampling bias at play here because the soft-bodied fossils were a bit of a surprise, and they were found late in the excavations at the site. And so they're very difficult to spot in the field. And so we're hopeful that more explorations at the Brecon site will reveal more of these soft-bodied fossils. Okay, so here's our specimen of this new morellomorph. We have a part and counterpart of the specimen, which we were able to prepare to show different details. And so I can digitally merge the part and the counterpart here and just show you what the whole specimen looks like. And so you can see that we have a pretty complete individual, a single individual for this new species, with most of the body parts here. So I'll go over the different parts in detail. But first, just to zoom in a little bit on the preservation of this particular specimen, we did some elemental mapping, and we found that the cuticle is preserved in a mainly carbonaceous mode. We do find also some evidence of calcium carbonate crystals, and you can see the calcium map on the lower right here, which appear to be sort of clustering inside the void formed by the carbonaceous cuticle. But these are very localized patches only in the appendages of the specimen. And so they seem to be sort of incidental to the overall carbonaceous mode of preservation. So just looking back at the large-scale anatomy of this specimen again, we see a couple of pairs of these really large ornate cephalic spines, which are lined with these arrays of secondary spines shaped sort of like fingers. We have this very large axial hypostome structure, which is pretty typical for these sorts of morellomorph arthropods. And this shows us that we're looking at this animal in a ventral view. We also have this really remarkable pair of hypertrophied appendages, which I've colored in blue here. And so we think that these are probably the second pair of appendages based on comparisons with other taxa that have a little antenule in front of them. And so aside from the size of these appendages, what's really interesting about them is they have this sort of flattened foot-like or tarsus-like termination, which we think has some ecological significance, as I'll discuss in a minute. We also found some other appendages in this specimen. So we found what we think is a third cephalic appendage, and then also a series of trunk endopods. Most of the trunk in this specimen appears to be missing, or at least we weren't able to prepare it. But we presume that, like other morellomorphs, it would have had a multi-segmented trunk with lots more of those trunk appendages. And then finally, we actually have some really nice small-scale details of this specimen. So if you zoom in on the appendages, you can even see these little pores, which we think were probably housing sensory setae. So again, we can come up with a pretty good reconstruction of this animal based on this single specimen. This is not bad for a morellomorph. These are a very rare group of arthropods. And we were able to reconstruct the animal as we think it would have looked in the Brecon environment. And so we think it would have used these really ridiculous long appendages to sort of plot its way across the soft mud in between the hard ground mounds. And in particular, these tarsus-like terminations on the hypertrophied appendages are really interesting because we suspect that they may have been used sort of like snowshoes to help this animal support itself on the soft, muddy substrate. Okay, so what kind of morellomorph are we looking at here? We suspected that this was a new species because it has a unique combination of characters. But where does it fit in the morellomorph tree? So there are two general groups of morellomorphs, the morellids and the acircostracans. And we're pretty confident that the new form belongs to the morellid group because it has these really ornate cephalic spines with multiple secondary spines that are characteristic of these animals. And that's as contrasts with the rounded carapaces that cover the entire body in the acircostracans. But we wanted to get a little bit more specific here with our taxonomy. And so we took to the literature. We came up with a revised character matrix based on a critical review of the literature. And I really have my co-author Alejandro to thank for doing a ton of work on this. We in particular were interested not only in placing our new morellomorph in the tree, but also testing the sensitivity of morellomorph in-group relationships relative to the outgroup hypotheses. And we were interested in this because morellomorphs have actually been one of the more controversial groups of arthropods in terms of placing them in the tree. And so it's not entirely clear what their closest outgroup would be. And so we took some parsimony and Bayesian approaches to looking at this. And we used this relatively small matrix, but by constraining the outgroup to represent different potential outgroup hypotheses, we hope to test the sensitivity of the topology to these changes in outgroup relationships. And so I'm just going to go over a couple of the key results. So firstly, the new morellomorph appears as the sister taxon to Mimetaster hexagonalis from the Devonian Hunstruck slate in all of our trees. And this kind of makes sense with stratigraphy because they're both our youngest known morellid taxa. We also recovered monophyletic Morellida and Acircostraca, but in some of our trees, we found this interesting result with pycnogonids clustering close to the morellids, which you might say is kind of crazy and surprising. There are actually some similarities here that this is something that we think still needs more work. And then finally, we find this relationship that's probably closer between chelicerates and morellomorphs and between mandibulates and morellomorphs.
Thank you, Joe. If you could wrap it up now, that would be great. Over time.
Yeah, so just to close, we found this new morellomorph. It expands our understanding of the diversity of morellomorphs, especially in the Ordovician. So it seems to be our northernmost known morellomorph. And we're hopeful that by further explorations at the Brecon site, we will find more material that can help us understand the biogeography of these sort of charismatic Cambrian groups better as we transition into the later parts of the Paleozoic. And so with that, I'd like to thank my collaborators and all of you for listening.
Excellent. Thank you very much, Joe. That's a really fantastic talk. If we can move swiftly on to Ross, who will be talking to us about constraining the origins of animals from taphonomic studies. Thank you, Ross. I can't see you yet, though. Excellent. Thank you. Take it away.
Great. Well, I am delighted to be here today at PalAS. And I first want to add my thanks to the organisers for enabling the meeting to take place at such short notice. I think they've done a great job. Okay. So the question I really want to grapple with today is really fundamental to the history of life on our planet. How old are animals? When did they first evolve? And it's a question that's puzzled scientists for a long time. Darwin famously lamented the lack of fossil evidence with which he could constrain the timing of origin of animal evolution. He wrote in The Origin of Species, "It is indisputable that before the lowest Cambrian stratum was deposited, the world swarmed with living creatures. But to the question why we do not find rich fossiliferous deposits belonging to these assumed earliest periods, I can give no satisfactory answer." Basically, Darwin was stumped. This was his dilemma. And today, a version of this dilemma persists. One line of evidence for the timing of the origin of animals, molecular clocks, which are built from the genetic data encoded in living animals, argues for an origin as far back as 800 million years ago. In contrast, the known animal fossil record maximally extends 574 million years ago to the fossils of the Ediacara biota. Now that's a discrepancy of over 200 million years, and it requires a scientific explanation. What is the truth behind the timing of animal origins? Well, there are two real possibilities. Either molecular clocks are simply inaccurate, or pre-Ediacaran animals aren't preserved as fossils. Now, if you've been to PalAS over the last 5 years or so, you'll have heard me talk more than once about the taxonomy of early soft-bodied organisms, including animals. And today, I'm going to combine the results of these individual studies to track fossilization processes across the critical interval from 1 billion years ago to 500 million years ago, in order to understand the extent to which the early fossil record can faithfully document the origin of animals. So to begin with, though, we first need to consider what the first animals would likely to have been like. How were they constructed? What features did they have that made them more or less likely to fossilize? Recent work using ancestral state reconstructions confirms that the earliest animals were very likely to have been entirely soft-bodied and to have lacked the hard shells and skeletons that usually preserve as fossils. So where do we go to preserve early soft-bodied animals? Well, Joe's talk has given us a nice introduction. The obvious answer is Cambrian Burgess shell-type deposits. They're renowned repositories for soft-bodied fossils, preserving a spectrum of animal tissue types in fine-grained siliciclastic rocks. Among the modern animal phyla, there are only 5 absentees from these famous deposits. Not only that, but they also preserve microscopic animals too. So if the first animals were small and soft, Burgess shell-type deposits would be the place to find them. So given that Burgess shell-type deposits might be the place to look, the taphonomic studies I'm going to focus on today will focus on 2 questions. Firstly, what are the conditions necessary for Burgess shell-type fossilization? And then secondly, do Neoproterozoic fossiliferous deposits share these conditions? So let's take the first one first. A large raft of work is going to understanding Burgess shell-type taphonomy with various hypotheses put forward, everything from limited oxygen supply through to the composition of the host sediment, through to the mineralization of fossils, through to the sealing of fossiliferous beds by orthogenic cements. Now, over the course of the last few years, I've become increasingly interested in the roles played by clay minerals, since they've been implicated multiple times, either through a role in the host sediment or by attachment or precipitation directly onto decaying carcasses. And the first taphonomic study I want to highlight today explores this relationship. So in 2016, we showed with a microbiological approach that some clay minerals, specifically iron-rich berthierine and aluminium-rich kaolinite, have antibacterial properties, providing evidence of a mechanistic link between slower growth of bacteria, suppression of decay, and the promotion of fossilization. Other studies, for example, studies by Nick Butterfield, have argued that clays may bind to organics, increasing their decay resistance. So the second study I want to highlight, which is one I led, showed with X-ray diffraction that these decay-suppressing minerals were common in rocks hosting Cambrian Burgess shell-type fossils, specifically the clay mineral berthierine. In fact, clay mineralogy of the host rock can predict with around 80% accuracy which Cambrian rocks preserve soft-body fossils. Earlier this year, I continued this work, publishing a study where we developed our X-ray diffraction methods to show that the precursor clay to berthierine, kaolinite, is directly associated with fossil soft tissues, suggesting clays not only played a role in the general sedimentary environs of the carcass, but also directly bonded with decaying tissues early in the fossilization process. So these first 3 studies show that clay minerals are a major factor in Burgess shell-type fossilization, playing multiple roles in the preservation of animal soft tissues. The early animal fossil record then is therefore likely biased to settings where clay production was favored. So now I want to ask, do Neoproterozoic fossiliferous deposits share these conditions? Is Neoproterozoic soft-bodied fossilization the same as Birdie shell-type fossilization? And what implications does the answer to this question have for how the fossil record can chart the origin of animals? So I've been working with Susanna Porter and her student Tina Waltz at UC Santa Barbara, and we've begun to test the association between clay mineralogy and the preservation quality of Neoproterozoic organic walled microfossils. Tina has quantified the preservation of these soft-bodied fossils whose fossilization style resembles Birdie shell-type fossilization on a scale from 1 to 3, with 3 being good, based on various metrics such as the pitting in the cell and the cell wall margin. We've again then looked at the mineralogy of the rocks hosting these fossils with X-ray diffraction. And interestingly, unlike with Birdie shell-type fossilization in the Cambrian, there's limited evidence for association of good fossils with sediments composed of specific clays. The charts I've shown here show the probability of getting the best preserved fossils with different clays for 2 quality metrics, pitting and cell margin. So the fossiliferous Neoproterozoic deposits we've investigated do not share the precise sedimentary composition of Cambrian Birdie shell-type deposits. And this is actually probably not all that surprising when you consider the decay resistance of the organisms preserved and their tissues. Microorganisms like green algae had cell walls that were relatively decay resistant, whereas early animals were composed of relative decay-prone tissues. Perhaps the organisms preserved in Neoproterozoic deposits didn't need such specific conditions to fossilize as early animals. So, do Birdie shell-type conditions exist at all in Neoproterozoic rocks? Are Neoproterozoic rocks capable of this exceptional style of fossilization that is so key to recording early animal evolutionary history? Well, to ask this question, I delved deeper into 3 Neoproterozoic deposits with some of the best preserved microfossils. And using a complex technique of focused ion beam preparation, followed by elemental and mineralogical mapping at the synchrotron, I documented kaolinite associations, even halos around the fossils, with some Proterozoic microfossils across phylogeny, from green algae to cyanobacteria. And this is just like we've recently found in Cambrian Birdie shell-type deposits. Kaolinite is directly associated with the tissues. So, returning to the first question I laid out, how old are animals? What do these taphonomic stories together tell us? Well, I think I've shown 3 things. Firstly, kaolinite and berthierine are key factors in Birdie shell-type fossilization, playing multiple roles, either in the host sediment or directly attached to the tissues. Second, most Neoproterozoic fossil localities don't share Birdie shell-type mineralogies or have Birdie shell-type fossilization conditions. And this second point is really important because it's the first evidence-based explanation for the disconnect between the molecular and fossil records. But the third point is some do, suggesting the conditions do exist, albeit very rarely, for the fossilization of Neoproterozoic animals. But let's take a deeper dive into the fossils preserved in these rare Neoproterozoic settings, mostly in the Tonian from the ones we've looked at so far, that do exhibit Birdie shell-type mineralogies. None of the fossils in these deposits could be argued to be an animal. And that's the key thing. Given that we have rare Neoproterozoic deposits with the conditions for Birdie shell-type preservation, but no animals, it may suggest that animals really weren't there and may provide a useful maximum constraint at 790 million years ago. That's the youngest age of the deposits we've investigated that do show Birdie shell-type conditions in the Neoproterozoic. A useful constraint at 790 on our molecular clocks. So we can plug this into molecular clocks and get a better calibration point. Now, there are likely to be some objections to this logic. Animals were hiding away from areas of preservation, or we haven't considered all taphonomic windows. Animals may be better preserved in cherts or phosphatized settings, or ediacara style sandstones. And I agree with these objections. These and others are all valid concerns. But for constraints on molecular clocks for calibrations, all we need is a probability. And the work I've presented here crucially helps to refine that probability. So in terms of Darwin's dilemma then, I hope I've demonstrated that taphonomic studies have real potential to finally provide the satisfactory, evidence-based answer he craved. So with that, I'll thank a bunch of co-authors and institutions that have really helped on these projects and say thank you.
Thank you very much, Ross. That was a really fantastic talk and very, very interesting work. And as we're all very aware, very important to try and get these evidence-based constraints on molecular clocks. Thank you. Right, so if we could move on now to Sebastian. Excellent, if you could share your screen, who's going to be talking about soft-bodied fossils from the Lower Devonian and how they can be used to understand the early stages of vertebrate evolution. Oh, we've lost him. Sorry. Sebastian?
Yeah, I'm there.
You could share your slides.
Yep. It's okay?
Fantastic. Take it away. Thank you.
Okay. Okay, thank you. Good morning, everyone. It's a pleasure for me to be there today and to share my research with you. My name is Sebastian Olive and I'm postdoc at the Royal Belgian Institute of Natural Sciences in Belgium. And today my talk will be on the project that started quite recently this year, and I will present the preliminary results. The title of this presentation is How Soft-Bodied Fossils from the Lower Devonian of Southern Belgium Can Help to Decipher the Early Stages of Vertebrate Evolution. So to carry on with my talk, here is a brief reminder of how recent chordates are defined. So vertebrates, urochordates, and cephalochordates are all chordates, as you know, and chordates are deuterostome animals that display, among other characteristics, an autochord in brown here, number 2, dorsal neural tube, number 3 in green, pharyngeal slits, number 10, and the post-anal tail here. Early chordates and early vertebrate fossils provide our only direct information on the origin of vertebrates and on how their distinctive body plan evolved. Unfortunately, the fossil record of early chordate is extremely scarce, as these organisms mostly consist of decay-prone soft parts that are usually degraded and lost prior to fossilization. That makes interpreting their anatomy highly challenging. And consequently, and Rob showed that in 2010, their phylogenetic position is unstable. For instance, Picaia in purple here has been interpreted as a cephalochordata, also as a lamprey, and even as an annelid, sorry, but it is not shown in this figure. Here are some examples of presumed early chordates. They are all Cambrian in age and come so from Canada and China. My postdoc project focuses also on another group of deuterostome, the Ephanerophids, that are a group of early vertebrates. Their phylogenetic position is not clear, as you can see here, but this is an interesting group since Ephanerophids are the first vertebrates that demonstrated the presence of gill filaments enclosed by gill pouches, paired anal fins and pelvic fins, and also an intromittent organ that has been shown recently by Chevrier et al. So here, for instance, a Phanerops from Canada with a bronchial basket here. You can see the gill filaments enclosed by gill pouches here. So thanks to phanerophids, it has been possible to decipher the condition of bronchial breathing within vertebrates. So, but talk about my fossils. The idea of my postdoc project started with these two awkward fossils. The one on the left was found in the paleontological collections of the Centre Grégoire Fournier, which belongs to the Marais-Sous Abbaye. We have the part and the counterpart, and the other one on the right belongs to the collection of the Royal Belgian Institute. And both come from the Ardennes Allochtone here in southern Belgium, and they are both lower Devonian in age. So our first thought when we saw the fossil from the Centre Grégoire Fournier was that it exists a striking similarity with early chordate from the Cambrian of China and Canada. So with the putative dorsal notochord that you can see here and the elongated shape of the body. But more details are needed to clarify the assignment of these fossils. Also, the second specimen from the Museum of Brussels, the putative bronchial basket, displays strong similarities with the bronchial basket of Euphanerops, because both are covered with dorso-ventrally elongated rod-like structures. But like the previous specimen, we need more details for those specimens. So to retrieve as much anatomical details as possible, we used and are using an array of classical and more innovative techniques. Among them, the bandpass emission macroscopy and the synchrotron-based micro X-ray fluorescence major to trace elemental mapping. Here with different fossils, just to show you the possibilities of the methodologies. So preliminary results. Here are SEM images of several areas of the bronchial basket. Colors around the same images correspond to the different close-up, yellow for yellow, etc. You can observe here the very delicate preservation of the bronchial fibers. The latter are enclosed in gill pouches that are partially preserved here. You can see one here. So the specimen displays two types of preservation highlighted by this interpretative drawing. These two types of preservation are also revealed by the synchrotron result and the distribution of the various chemical elements. We don't know exactly to what corresponds this different preservation, maybe simply to a different view of the bronchial basket. Here we would have the external view of the basket, and here the internal view of the other side. Regarding the specimen from the Centre Grégoire Mounier, here you have the results for the synchrotron. I will discuss the interpretation in a few slides. And here, the bandpass macroscopy results for the same specimens. So we have the entire fossil here, and here only the counterpart, but only the anterior area. So preliminary results, we can see that there is a different structure of the anterior part of the body compared to the posterior part. It argues for a cephalization, so with the head here and the body here darker. Two rounded black structures are in front of the head that could be interpreted as eyes. On the anterior part of the head, we have elongated structures that could correspond to head appendages. The notochord is clearly observable here in red with a synchrotron. We have dorsal processes like a segmentation with maybe what is a nerve cord here, pointed by the yellow arrow. We have also different fins. The dorsal one, which starts at the anterior part of the body and stretches until to the middle length body. And we have here, it's clearly visible, ventral fin that goes all the length of the animals, along the ends of the animal. We still have some patches that we do not understand with white stars. Maybe this one corresponds to the brain. We still have to understand what we observe. So preliminary conclusion for the bronchial basket. Here is a summary of what I said, but this specimen is not connected with the rest of an animal. So the phaneropid assignment should be verified. For this, histological cross-section in the rod-like structures could help to attest the presence or not of calcified cartilage as in a phanerops, as shown by Philippe Janvier. For the putative early chordates, is that truly an early chordate? Could that be anything else? So even with modern and cutting-edge technologies, the interpretation of enigmatic fossils remain difficult, and more specimens are needed. And that's a chance because very recently, we likely found the original quarry of this early chordate specimen. We spent only 2 days with a team of roughly 10 people and found more than 100 specimens of early chordates, vertebrates, and invertebrates. Here are examples of new soft-bodied chordate collected. The preservation is really different. We have to study that very soon. Some of them are periodized, as you can see here or here, and other ones are observable only by traces. So this for early chordates, putative early chordates, and here for the different fossils we found, vertebrates with probably Drepanaspis here, a shark spine there, and different invertebrates. It's possible to identify some of them, but if you have ideas on other specimens, I would be very interested because I don't know. This locality could be an equivalent of the famous German locality of Hunsrück.
Sorry, Sebastian, you're running out of time. If you wrap it up now, that'd be fantastic. Thank you.
Acknowledgement to the PalAS Organization Committee and all the different partners. Thank you.
Excellent. Thank you, Sebastian, for a really fantastic talk.
You're welcome.
If we could move on now to our final talk of the session, Carlos,
yes,
talking to us about conodonts. Good morning.
Coming.
Share your slides.
Yeah, should be fine now. It's coming.
Excellent. Thank you, Carlos.
Okay, you can hear me.
Right.
Okay. Yep,
yeah,
sure.
Yeah, that's all good. Thank you.
So thank you. Can I start?
Yes, please do start.
Okay. Yeah, so I mean, I'm not sure if this is related or strictly related to special preservation, but at least the last talk is good of the session. So I'm Carlos, and I'm going to try to talk about the physical characterization of a specific tissue that is found in the conodont corn tissue. Obviously, there have been several talks about conodonts during the conference, so I'm not going to come back to the introduction, what's a conodont, affinity, etc., but just to focus on a few points that, as probably you already know, the fossil record of conodont is basically comprised by these isolate elements that originally were part of a complex apparatus. But the important thing is these elements, beside the affinities or not belonging to vertebrates, is that they composed by phosphate, by hydroxyapatite, a tissue that is just very common. This is common in vertebrates, and of course has a specific features for the fasciogenesis that allows us to use for different studies. As you know, conodons were important part of the ecosystem in the past for more than 300 million years. So one of the important things, or what's basic on this work, goes that conodons have been used for a long time for for correlation and ages the rocks where they found. But the other important big controversy was around the affinities, if conodons were or not vertebrates. And of course, one of the main points was related with histology of conodons. When we compare conodons tissue with a vertebrate tissue, are very, very similar. We already know that they are not homologous. It's a convergence, but they still have another features in the soft body, in the soft tissue that we found in the body preservation in Scotland that can help us to understand the vertebrate affinities. But the thing we see is this: you have a very hypermineralized tissue, very similar to enamel and dentin. And basically conodons, we can see that they have these two different parts, very similar to what we can find in vertebrates, the crown tissue and the basal tissue, basal body, that are not exactly dentin and ameloid, but they are very similar. The interesting thing with the crown tissue, when we saw them in transmitted light, it's very easy to find that they have two different tissues just built in the crown. Is the so-called the hyaline tissue is translucent in microscopic light, or the albite or opaque white matter that you see that's going to be our target in this work. And why? Because this tissue is specific for conodons, as we can see when we do histology. Is completely different to the enamel-like tissue that we found in the crown. It's characterized by these growth lines and this prismatic appetite growing, very similar to the enamel invertebrates. And this white matter tissue, this is the one that we can see in this part of the slide, that is very difficult to see growth lines, is very massive, apparently, and is always characterized by this porosity or these voids in the tissue. Interestingly, we can see that this tissue is distributed mostly in the core of the different denticles in so many conodons. It's not always present, but when it's present, it's very common to find in this part. So the interesting thing is, because it's a specific tissue of conodon, for the last 20 years or more, several groups have tried to characterize the nature of this tissue. One of the most important ones in terms that they try to is an important work trying to describe the different tissues with new techniques was Donahue, and he couldn't resolve the crystal size of the world matter, probably because of the technique used. Later, Trotter, using transmitted electron microscope, describe this crown tissue or this white matter tissue as a single crystals, the hundredth of micrometer in length. But the more interesting thing, the more recently using another technique like electron backscatter diffraction, Perez Huerta couldn't resolve any crystal, so getting the conclusion that this tissue was microcrystalline. So it's very confused. So different techniques, different authors trying to see what's going on, and at the moment, it was difficult to say that who was right. But the important thing in this is that conodons, because of this hypermineralized tissue, are the target for geochemical studies for a long time, because this apatite is very stable. So we can think that the original isotopic signal is still there, so we can use for paleoclimate reconstruction for past ocean chemistry, temperature, etc. So it's very common to use oxygen isotopes, try to describe how the temperature in the past oceans change. The problem is that we have seen that this tissue is very porous, and if we have a tissue that is very porous, it could be very permeable, and then it could be very important in geochemical changes in the original signal. So probably we need to be sure that this tissue, or the crown tissue, is still suitable for geochemical archives. So basically, our objective is to attempt to resolve the nature, porosity, and permeability of this specific tissue, using tomographical techniques and helping by pore network analysis and combining with electron backscatter diffraction. This technique is to try to help us to see the crystallographic nature of element. To do that, we just focus on a couple of taxa, basically Othelcodina confluens and Pteridontus nakamurae, basically because they was privileged used to describe this tissue, but probably most important because we can see, and that's what they used it before, is because this white matter tissue is very well developed in the inner parts of the different denticles. So as said, to try to characterize it, we used two different techniques. Was tomography. For that, we went to the Synchrotron de Pulsar Institute in Switzerland, and we used two different detectors, the Tomcat and the CSAX, basically trying to resolve different points. The main important difference between these two detectors, both use tomographic X-ray tomography as a basis, but the resolution that we can get in the two detectors is completely different. We can get to nanometer the resolution. With this, actually, we try to combine both the general distribution of the tissues using Tomcat, and then go into specific part of the conodon, because this CSAC has very important constraint with the size of the sample. We need to go to less than 100 micron size sample, so we just focus on the tip of the conodons. As I said, we combine this with electron backscatter diffraction, just trying to provide quantitative microstructural information about the nature of the white matter. As I said, this technique was used before for trying to describe the conodon tissues, but Pérez Huerta within it fails. And basically because it's very complicated technique for appetites. And the proof was that I said there was the co-author that's working with this part of the manuscript. It took almost a year to get results. So he needs a lot of time just to get the specific protocol to try to get the results. I will show you later. So the tomographic, the results are here. So basically it was a video, but the code wasn't working. So we get the highest resolution probably for a fossil with a Boswell size of 44 nanometers. But it was good in terms of he allowed us to recognize in the resolution the different voids and pores through the specific volume in the case of Pteridontus. Again, you can see in the Tomcat that this tissue was distributed along the cast, and it was completely different from the laminar tissue that we can see in the base of the element. So as you say, it's a very porous material. So I think that our doubts about its porosity, I think is easy to understand. Then in order to visualize this and say, okay, if we have or not, this porosity could have important or affect importantly to the permeability of the element, we conduct a pore network model using the extension provided by the VISO, basically to visualize the volume of the different pores and the connections between them. As you can see in this picture, it's not ours, it's just the example that the VISO gave you. So doing that, we measure a specific part, a small volume of the cast, giving us as a proxy that 0.5% of the volume was represented by these pores. And we have thousands of them in just in a very small volume. But the interesting thing is that we, for the first time, could see how this porosity was distributed, three-dimensionality in the tissue. And we saw that, of course, not of course, wasn't clear, but the biggest voids were in the center, and they just were reducing size. But as you can see, basically, it's not a reduction on the side; it's just a very homogeneous size of the pores from most of the extension of the cast. But the most important thing, as we can see in the top right, is that we could see how these voids were arranged in growth lines parallel to the external surface of the element. So these voids were adding during the growth of the element. The most important thing here is that the voids are not connected between them, and neither with the exterior. So that's the more important thing that we found with this analysis. Finally, we ran this electron backscatter analysis diffraction just to see if this white matter how it behaving crystallographically, and we finally found this every single cast was just filled by a single crystal of apatite. We saw in Othelcodina, but as well we saw in Pteridontus, the specimen we studied with the tomography. That's very, very interesting because very important for us. So there's important conclusions. The first one is, of course, that this the tomography And the pore network analysis allows to characterize this tissue and allow us to say this is not permeable, since pores are not connected between them or with the exterior. But given extra information about how this tissue grows, because there was so many controversies about it, and the presence of this porous in concentric lines parallel to the surface. Okay, this one is like extra. Parallel to the surface is just is congruent with this element or this material grows syntactically in crystallographic continuity. That was very important because there were so many doubts about how this tissue could grow. But again, we saw that, as Trotter said, we can just confirm the Trotter's conclusion that this tissue just behave or is a single crystal, as a macrocrystal, not as field-cooled, resolved, or Perez Huerta. So in conclusion, it's in terms of the physical characterization that we can say that this tissue can be used for geochemical analysis. But an X-ray is that this tissue is specific for conodons. And we interpret it as a very sophisticated tissue for the function that they develop as teeth. Why? Basically, because it's a specific tissue, very, very strong tissue, yes, in the cusp or in the inner part of the cusp, that give an important stress or compressive stress to the animal. But it's a very homogeneous. So the presence of this inhomogeneities, this porosity, is probably part of an adaptation to the cusp propagation cracks. So we don't have different decussation planes, but we have voids that in the even propagation or presence of a crack propagation, they will stop and avoid the loss of part of the element. So at the end, besides the utility, we can as well conclude that probably what matters is the very, very sophisticated adaptation for the codonon function that they could develop. So thank you for your attention.
Thank you, Carlos. That was a really, really interesting talk. So now we've got 13 minutes of questions and answers. So all of our speakers have given fantastic talks and been very good at replying to the answers on Slack. But we thought we'd now discuss some of them for those of you who haven't been able to keep up with Slack. So Morten, you had a few very interesting questions. So do you want to talk a little bit about the extent to which diet impacts the likelihood of phosphorylation, and also whether there's a correlation with the thickness of body walls?
Yeah, sure. Diet might be another thing that enhances phosphorylation. That would make sense because if you are a carnivore, then you ingest a lot of muscle tissues and stuff which are phosphorus rich. And therefore, you would increase the amount of total phosphorus within yourself. So that might be another factor that we see. And sometimes in the arthropods in serious passage, we see that the gut tracts are only phosphatized in local patches. And perhaps that could be due to the location within the gut of ingested food. However, it's a bit difficult to find any remains because these are very mineralized masses. So it's a bit difficult to test, but it is something that I want to look further into to see that. And the other question, whether taxa with thick body walls would be able to contain the phosphorus within themselves. Creating a barrier to the external environment is important, which was shown by the enrolled arthropods, which favored a more extensive phosphorylation. But we still see some arthropods, and even one species which also enroll, which do not phosphatize anything, despite that they have these diverticulae, which were supposed to be phosphorus rich. And then we have the stem ketogaths, which have body walls which are supposedly thinner than these arthropods, and they still phosphatize around their ventral ganglia. So I think that body wall thickness might be a might be effects, but I don't think it's as important as, for example, phosphorus content. It's also possible that it's not about phosphorus diffusing, but it's about the nucleation of phosphatization in these phosphorus-rich tissues, which then extends into the adjacent.
Excellent. Thank you, Morten. Yeah, really interesting stuff. Okay, so if we can move on to Alison. So Alison, I guess the two questions were kind of, well, to start with, do you think other cephalopods such as Phaeocipio should be included in your phylogenetic analyses? And are all the soft tissues preserved in the same minerals within your specimens?
Yeah, so great question. Phaeocipio is controversial, and yeah,
sorry, I don't know much about cephalopods. Could you explain a bit why it's controversial?
Sure. So some people have positioned it as like the earliest stem octopod, I believe, if I'm correct, and it's from the Carboniferous. But there's questions about that. There's poor preservation. I think we're missing the internal structure, and also the addition, I think, of 10 arms. So there's question about that. And I know Tanner 2017, I think, they did a molecular clock analysis, and it's not octopods aren't supposed to have originate before the Triassic. So yeah, there's questions about it. For our phylogeny, we did include it. It was in the Kruta et al. 2016, and previously in Sutton 2015, which is what the Kruta et al. Was based on. So we did it included in ours, but I know that it is sort of a controversial person. And then regarding the mineralogy, we didn't really do analysis on that. We were just really mainly focusing on the morphology. But we know that the soft tissues of marine arthropods that were fossilized at Lavault, we can see that the muscle tissue is replaced with fluorapatite and pyrite and sort of the related sulfides. So we're just assuming the same for Vampironassa in this case.
Excellent, thank you. That's really interesting. If we could move on to Joe now. So unfortunately we overran a bit, Joe, and you didn't have enough time to talk about your very exciting results. So I was wondering if you just wanted to use a minute or two to expand a little bit on what you might have said if we'd had more time.
Well, I think I got most of it in, but I guess just to finish up, I'd say we're really excited about this discovery of new soft-bodied forms in the Ordovician of Ontario. This was something that was really unexpected, because we don't tend to associate these things with being preserved with a whole huge diversity of shelly forms on these carbonate platforms. And so we are really hopeful that the further exploration of Brecon will reveal more fossils like this, but also I think beyond Brecon, looking at similar sites, in the Cincinnatian, for example, there could be potential for these sorts of things to turn up there as well. So maybe we just need sort of the right search image for it.
Yeah, no, that would be really exciting. So with regards to your results, Marielomorphs as stem picogonids, that'd be very exciting. Could you expand a little bit on kind of what you think the implications for that might be?
Yeah, so that's it's something that seems a bit crazy when you first think about it, because these two groups are very different from each other, and they're both very weird and sort of problematic forms. In terms of the characters that unite them, though, we found some similarities in terms of the differentiation of the head appendages, the very highly segmented large appendages on the head, the lack of lateral eyes in most of these taxa. So there are some characters that potentially unite them. I think we should be a little bit cautious about this result, just because this particular matrix that we created was really designed to test in-group relationships. And so we weren't necessarily focused on testing the different out-group hypotheses, but more trying to control for sensitivity in the in-group relationships. But in terms of implications of the finding, I mean, it's quite interesting in that morellomorphs have long antennules, whereas pycnogonids like euchelicerates have their characteristic little chelicerae, their pincers at the front. And so is this suggesting that chelicerae, for example, could have evolved convergently? I think we need more work, but
yeah,
it's definitely intriguing.
Very intriguing. Excellent. Thank you, Joe. If we can move on to Ross now. Really interesting and important questions that you're addressing, Ross. So one of the questions you got asked was about chitinous microfossils, such potential fungi, and and indeed more generally kind of incorporating different sorts of taxonomic modes into your analyses. Do you have any kind of, how much of this are you doing, and what do you have any predictions for what effects they might have?
Yeah, so on this question about whether we'd looked at clay-organic interactions in fungi, not so far. What I would say is that we're just touching the surface on actually probing the underpinning mechanics behind interactions between clays and organics. I think there's a lot more that we can do on that. In terms of the specific microfossil that was referenced, working with the microfossils is challenging. And so some of the techniques we've worked on are essentially unique in that it requires a lot of preparation of the fossil to work. And so we've only done a very few microfossils so far, maybe I think 5, but we've looked at cyanobacteria, green algae, and then other kinds of Incertae sadis microfossils. So yeah, it'd be great to work on that, to look at that.
Excellent. Thank you. I'm sure the discussions will continue in the chat.
Yeah, Emily, can I make one quick point? Is that possible?
Sure, go for it. Yeah.
So there's this debate between Graham Budd and Phil Donoghue on the Slack about use of calibrations, and I think this is where taphonomy can play a really big role, right? How we choose those calibrations on the molecular clocks is really important, and at the moment we're picking them based on different folks' interpretations. And the problem is we don't have a good way to look at absence, right? When a fossil's not there, is it really not there, or is it not there, right? And so I think this is where taphonomy can be really important and can actually help us on this regard, because it can help us have an evidence-based way to discriminate between how we choose these calibration points. I think there's a lot of potential there for its use. And one of the things I'd really like to do is look at mineralogy survey mineralogy across a billion rocks, 500 million years ago. And we're planning to do this to try and track these conditions through time, see if there are intervals where they're more likely to be there or not, to see if we can put some probabilities on when these fossils are most likely to be able to be preserved or not. I think it's a lot of potential in that technique.
Yes, yeah, no, that'd be really fantastic to try and quantify the probabilities, as you say. Thank you, Ross.
Thank you.
So Sebastian, if you're willing to speculate a little bit, what do you think the head appendages for your fossils might be?
Yeah, if they are truly head appendages, they could be used, for breathing. If there are gill elements on their part, but we still have to look in the details for that.
Okay. And do you find any skeletal elements associated with your putative chordates?
Yeah, in the locality we have shark spines and Drepanaspis plates, but apart those specimens, no, we don't have skeletal elements.
Okay, excellent, thank you. So I think we're running out of time, so we're going to move on to Carlos. Carlos, was the white matter exposed in the functional surface of the conodonts?
I think I was just answering Mark. I think so, yeah. I'm not sure I was said him in this specific element we studied because I didn't go through them in very deep detail, but in other specimen we scan, I will say yes, this at some point should be exposed because we found marks of wear in this thesis. But as Mark was saying, of course, we have a problem of how they could grow. But I'm probably saying that this repair process is basically, at least in the specimen that we scan, that we can see this in 3D very, very well. It's a project we are running with Emilia and other students, is that probably the lamellar tissue is repairing and recovering these denticles, but the core of the denticle is still with white matter, and the microwear just reach this, or the wear facets or problems could reach this material. I think so. I still reconcile how they grow. Phil is much better than me on that, so he will help me to find the solution.
Excellent. Thank you, Carlos. So unfortunately, we are out of time, but I'm sure if people ask their questions in the Slack, our speakers will continue to answer them wonderfully. So all that remains is me to thank all the speakers for really fascinating talks this morning, and thank you for attending. And I'll conclude the session and look forward to